Good morning, everyone. Welcome to the second day of this course, Brain Health for Life, not pretending, but preventing brain-related disability that we have the opportunity to hold thanks to the support of BioCat, of La Caixa, of the Institut Guttmann, of the Universidad de Barcelona, of my own home institution in Boston, Harvard Medical School and Hebrew SeniorLife. We're very excited about the course so far. The day yesterday we feel was quite interesting. We learned a lot thanks to the talks by fantastic faculty about different aspects, different key lifestyle factors that contribute to sustaining brain health across the lifespan. And today we are carrying on with a slight bend in the topic, looking at the concept of resilience, of reserve, of maintenance, and how that plays out and interacts with these various lifestyles and what we can do to modulate it. And given that focus on promoting brain resilience and reserve, nobody better to get us started than Professor Yaakov Stern. It is my great pleasure to introduce Yaakov, who will give a keynote sort of speech up front and then moderate the session for all of us. He's also chairing a very important initiative supported by the NIH to try to get some clarity on these very key important concepts of reserve, of resilience, of maintenance that we all use, and that we heard yesterday from one of the speakers. We all feel like we know what they are, even though it's hard to define it. Well, it is hard to define it, and we need to understand better what we mean with it to make it really fully useful. And therefore, we look forward to learning from Yaakov and all the other speakers this morning. And so without more delay, let me pass it on to Professor Stern. Thank you very much for joining us.
Hello, everyone. It's my pleasure to be here. Let me bring up my slides. First test. Here we go. Okay, so the title I was given is Promoting Brain Resilience and Reserve, something I've been interested in for a long time. I should point out that all the work that I do is supported by the National Institute on Aging. And this is something new I like to put up, that Academic Commons at Columbia has reprints of papers that our group publishes. Okay. So as Álvaro mentioned, because I'm gonna mention it several times across the talk. Álvaro is a co-chair of this along with several other people. The NIH, National Institute on Aging, has supported this collaboratory on research definitions for reserve and resilience. The idea is really to get people to come to consensus on definitions of these various concepts that we all use: reserve, resilience, brain maintenance, etc. And the program, as demanded by the NIA, has several components: 3 workshops, work groups, data sharing, pilot studies, and dissemination. And I wanted to raise it now because I'll be mentioning this collaboratory at several points across the talk. I'm gonna talk about 3 mechanisms underlying resilience. You notice I'm using resilience as an overarching term: brain reserve, brain maintenance, and cognitive reserve. Brain reserve in certain ways has become a controversial term. I didn't make it up. This was proposed by Robert Katzman many years ago. He was doing a longitudinal study of aging in the Bronx, that's north of Manhattan, where I am. They had a big community study, and he wrote an interesting paper where he described 10 women who were cognitively normal at death, but had a lot of amyloid plaques postmortem. And they were developing this idea that the plaques correlated with cognition quite well, but here were these people that had a lot of plaque were cognitively normal, and they speculated that they started with larger brains and more large neurons, and this might be said to have greater reserve, and introduce this concept of brain reserve. The idea that there's more neurobiological capital, more neurons to lose, I think is what he meant. This concept's a bit interesting and controversial now. I'll get to it later, but I think I could suggest that at any point in the process of brain maintenance is a longitudinal process, but at any point, someone might be said to, because of what they maintain, might be said to have a certain amount of brain reserve. And I'll leave it at that for now. Here's a very interesting early paper. Peter Schofield now is in Australia, but was a postdoc in our lab then a long time ago, and he convinced people in our community study to measure head circumference. And here you have head circumference from smallest to largest. Here you have prevalence of Alzheimer's, and you can see the smaller the head, the larger the prevalence of Alzheimer's disease. So that's brain reserve in its simplest. Now, cognitive reserve, what I'm showing you here is a new definition that is worked out for the collaboratory that I mentioned. I think it's longer than definitions we've had before, but I think it really is, I like it. So cognitive reserve is a property of the brain that allows for sustained cognitive performance in the face of age-related change and brain insult or disease. And then this property of the brain is defined. It could be multiple potential mechanisms, including molecular, cellular, or at the network level. Sustained cognitive performance, we want to think about trajectories measured longitudinally, although cross-sectional data could be illuminating. And these mechanisms could be characterized via biological or cognitive approaches. Then we added on another potential mechanism through which reserve operates is the reorganization of functions, sometimes referred to as compensation. And that refers to imaging studies where we can look at the efficiency or capacity or reallocation of resources. But that's one potential mechanism, but there's numerous others as well. So that's cognitive reserve in a nutshell. This is an old figure. This is Satz. Paul Satz is a neuropsychologist who wrote a very interesting article on brain reserve capacity. And he talked about two patients, one with more brain reserve capacity than the other. We could call that more neurons, more cells, more brain. And he posited a functional impairment cutoff. And so if this patient too has less brain reserve capacity than this one, they both get the same lesion, but this person starts to have deficit because it's pushed them below the functional impairment cutoff. And I call that a passive model. So because we're not talking about what the rest of the brain is doing, we're just talking about the amount of capacity. And if you go below that amount of capacity, all of a sudden you have deficit. And I would say brain reserve is that type of a passive model. And cognitive reserve, I'd say, is an active model. And that's an important thing to think about. So you have two people with the same brain reserve capacity. And today, I would say, you measure everything about them, cortical thickness and white matter tract integrity and how many plaques and tangles they have. They're equivalent. And then, well, let's talk about their cortical thickness. Then you have some hit to the brain like plaques. And this person can tolerate a lot more than this one before he or she starts to show deficit. And that's because the brain is actively trying to compensate, to deal with, to cope with that damage. It's an active process that allows this person, even though they've lost more of their brain reserve capacity, to be able to continue operating without functional impairment. Now, that idea that it's an active process, I think is really important. It's not something that you start with, so if you have more of it when you start with, you can tolerate a lesion better and it's worn away. No, the brain is actively coping with, and I'll come back to that theme a few times. Here's the earliest paper I ever published on cognitive reserve, although I didn't call it that in this paper. Here we looked at 20, 20, and 20 Alzheimer's patients, people with moderate Alzheimer's disease, mild to moderate, matched for disease severity in terms of mini-mental and function and all of that. And we looked at neurodegeneration. We didn't know that that's what it's called now, but now we know that this neurodegeneration as measured with PET is the final outcome of plaques and tangles and all of the other Alzheimer's pathology. And what we're showing here is that even though these people are matched for disease severity, the people with below high school education, HS is high school, have much less neurodegeneration than the people with above high school education. So that is, even though they look the same clinically, the people with higher education have more pathology underlying them. And we posited that that's something to do with education, allows some people to cope with more pathology than others. I like this slide a lot. So I think I've showed it at every talk I've ever given. It's a little joke amongst my friends, but I think for the purposes today, I think it makes an important point. This observation incorporates three key components for studying cognitive reserve. And I'm going to come back to these three components later. So the way I thought of them is, okay, I have a measure of the pathology or brain change, in this case, the neurodegeneration. I have a measure of the clinical outcome of that pathology. As I said, in this case, that's their mental status and their function. And I have a potential moderator of that relationship. In this case, it's education. And I think that it's important to keep all three of those things in mind if we're thinking about cognitive reserve. So basically what we're talking about is, okay, the brain is in a particular state. Cognition is at a specific level, and there's a moderator such that the people with higher reserve can cope with that brain change better and do better cognitively, or not become demented, etc. We'll come back to that in a little bit. Here's an early incidence paper where we showed that people with lower educational or lower occupational attainment were less likely to get demented. So the relative risk in the low education was twice as high as the high education. The relative risk of developing dementia was twice as high. And again, the assumption here is that if you follow people over time, on average, everybody has the same amount of brain pathology, but that the people with higher education somehow can cope with it better. Thus, that puts off the onset. And here's another early observation. This is just a schematic of it, where we found that in terms of, again, dementia, people with higher education or higher cognitive reserve actually have a more rapid decline. So if you think about it this way, you have increasing pathology over time. Let's talk about a memory test score. So the people with higher reserve will be affected by that pathology later. They've coped with it longer. And then what happens is they actually decline more rapidly because their resources have depleted so much that when it breaks through, they show this more rapidly. So these were very early observations about cognitive reserve. But again, what we're talking about again, we have pathology. That's the one side that's causing a change. We have memory on the other side, and then we have some moderator. In this case, we're talking about education. And then, you know, over the years, we've tried to understand what is the neural implementation of that cognitive reserve. What are some people doing differently than others that allows them to cope with brain change? And I think I should stress that. So here's a study where we are looking at a delayed match to sample task, a Sternberg task, where people see 1, 3, or 6 letters. You wait a while, and then they have to decide, was that one of the letters that I saw or not? There's a certain, the more the letters, the longer that decision takes to, time that decision takes to be made, so that with increase in this set size from 1 to 3 to 6 letters, we see increase in reaction time. And older people are slower than younger people very often at this. And what we can do is look at the brain, let's say during this retention period, and we can see changes in task-related activation as this set size increases. Now, we're making a big leap there. So we're saying, let's look in normal people, young and old, at load-related activation, and that can give us insight into cognitive reserve. So we made a leap from Alzheimer's disease to age-related changes. We've made a leap from people who may or may not show frank deficit to well-functioning people. And we're still positing that there's differences in underlying networks and efficiency of underlying networks. I would call this the slope of this increase in activation difference in efficiency. And we're saying that even in young or middle-aged people who are not demented, there might be differences in the efficiency of networks that might allow them to cope better with brain changes, just a standard age-related brain changes. And you know, we've actually worked it out. So here, this is this diagram comes out of many studies that we've done, and really is a mathematical path analysis, although I'm just showing it to you as sort of a theoretical thing. So you have age-related changes that do impact on task performance, and what happens in the brain. Either they reduce the efficiency of the brain, of the network. If that network doesn't work anymore, it might force them to use a different network to compensate with a different network, and that results in reduction in task performance. But what we found is that these various proxies of cognitive reserve, like education and IQ, maintain efficiency, reduce the need for compensation, allow people to compensate better, and thus task performance is better. So again, my point is not to make you fully, I hope you get a sense of what we're doing, but the idea is that the regular task-related networks, the cognitive networks, really might differ throughout life as a function of different exposures and allow people to deal with age or disease-related changes better. Now, the concept of brain maintenance, I think, came later. I'm sure Lars and his colleagues were thinking about it for a long time, but this is when I really learned about it in great detail in this article. And a different point, that individual differences in the manifestation of age-related brain changes and pathology allow some people to show little or no age-related cognitive decline. And so, at least in normal aging, the relative lack of brain changes and pathology is the biggest contributor to heterogeneity of cognitive aging. So that's another thing. That's not reserve against brain changes, that some people show less brain changes than others. That's a very, very important concept. And various genetic, environmental, and lifestyle choices could play a role in maintaining brain integrity and cognitive performance. So that's another side of the coin. Maintain the brain. If the brain is not maintained, perhaps you have reserve against that change. And there are two sides of the coin as far as I think about it. Here's an early paper from Michael Valenzuela. He rated people on this life experiences questionnaire, and he measured hippocampal volume over 3 years, and the people who had higher LEQ, more stimulating life activities, seem to show less decline in hippocampal size than the people with lower LEQ. So that's not cognitive reserve, that's brain maintenance. This is even more intriguing. This is the first paper I recall like this from Landau and Bill Jagus' group. They looked at PIB uptake, that's an amyloid PET, and they looked at Bob Wilson's cognitively stimulating activity scale and looked at midlife cognitively stimulating activities. And they found that those who engaged in more cognitively stimulating activities had less amyloid in their brain than those who engaged in more. Now, I never imagined that cognitively stimulating activities might impact on amyloid. And really, this is just sort of a convenience sample, and it may or may not be the case, but that's not cognitive reserve, that's brain maintenance. Okay, I don't know how that happened. So this is the collaboratory definition for brain maintenance. It's a lot easier. Brain maintenance refers to the relative absence of change in neural resources over time as a determinant of preserved cognition in older age. So for brain maintenance, everything is maintained, including the networks that underlie cognition. And this is what we added Lars did and say this, we did brain maintenance and cognitive reserve are complementary concepts. One refers to relative preservation of the brain, while the other refers to sustaining cognition in the face of brain changes. And both brain maintenance and cognitive reserve can be influenced by multiple genetic and environmental factors operating at various points across the lifespan. And I think that's important. And I just want to show you a study. It's not the greatest, but I think it's just a nice demonstration. This is my colleague, Chris Habeck. So the title of the paper was Cognitive Reserve and Brain Maintenance are orthogonal, but have common influences. So there's 3 steps. First, he established a measure of brain maintenance. It's not ideal because this was cross-sectional data. So basically, he looked at people's brain structure, 3 types of brain structure, and looked to see if people were above the average for their age, using a sliding window, or below. So this particular person is a little above the average for that age. So this person would be termed to have more brain maintenance. And he did this with 3 modalities: cortical thickness, cortical volume, and white matter tract integrity. Then he estimated cognitive reserve, again, using a residual approach. So he took a cognitive measure. He looked to see how much of that that measure could be accounted for by volume, thickness, and white matter tract integrity. And the residual he called cognitive reserve. So this is the ability for people to perform better than predicted by their structural status. And he did that for 4 measures, and he got a cognitive reserve variable. Much better, again, if we had done this longitudinally. But this is what I wanted to show. Now we correlate everything with everything. So take brain maintenance. It correlates with an estimate of IQ, the NART, and it correlates with education significantly. Take cognitive reserve. It correlates with the NART, again, maybe more strongly, and with education. But cognitive reserve and brain maintenance do not correlate with each other. They're both influenced by the same things, but they're 2 different entities, and they're each influenced in their own way by these lifetime activities. And I think that's a very important point to keep in mind. Now, the collaboratory was really a request from the National Institute of Aging, because we're working with these concepts, and the terms we're working with are complex, and people use the same words, same words for different concepts, or different words for the same concepts. And at the least, we need shared definitions that include how they are operationalized in research. So the goal of the collaboratory was to develop a set of well-accepted operational definitions and research approaches that can be disseminated to the field at large. And really important in that was to include non-human study, because we can only get so far in studies of humans to understand these underlying causes. But if we can get to animal studies, we can really get much more deeply into it. But we really have to make sure that we're all playing the same game and talking about the same thing. So we started with this white paper. This white paper was pre-collaboratory. It comes from the Reserve and Resilience Protective Factors PIA. This is part of the this is a group that meets every year at the International Alzheimer's, the Alzheimer's Disease Association Alzheimer's meeting. And 31 of us got together and wrote this white paper where we defined cognitive reserve, and brain maintenance. And we had consensus definitions for brain reserve, maintenance, and cognitive reserve. And we treated resilience as an overall descriptor for these concepts. We said resilience, that's everything, but we can back off and talk about brain reserve, cognitive reserve, and brain maintenance. And interestingly, and very importantly, I think the second part of this white paper provided research guidelines and operational definitions. If you're doing a longitudinal study, if you're doing an imaging study, how would you do it? And the paper points out the challenge of applying these concepts to animal or basic neuroscience research. So I thought this was pretty good, and probably maybe a good starting point for the collaboratory. But just to give you an idea of the rest of the field, here's a paper that Roberto Cabeza, who's also, along with Álvaro, on the organizing committee of the collaboratory, put out as a consensus paper from a meeting in Montreal that was really originally supposed to be focusing on how to define compensation. And unfortunately, I couldn't make it to the meeting, so I couldn't head. I'll be very honest, I would have tried to head some of this off at the pass, but maybe I wouldn't. This is a consensus diagnosis, consensus statement. So maintenance, which would call brain maintenance, is pretty much what we've been talking about. Reserve was backed off. First of all, they said, let's not call it cognitive reserve. It's just reserve. And this brain reserve is in the brain. Why make a difference between cognitive reserve and brain reserve? And reserve is really the cumulative improvement of neural resources that mitigates the effects of neural decline. So it's sort of like an intercept as far as I can tell. And compensation as cognition enhancing recruitment of neural resources in the response to relatively high cognitive demand. So here's a sentence that I think is interesting. Reserve is used to refer to the accumulation of brain resources, maintenance to the preservation of these resources by constant recovery and repair, and compensation to the deployment of those to task demands. From my point of view, what this does is it takes cognitive reserve and makes it into like a starting point, and tries to use compensation as the active process that occurs. I do feel that compensation is really an active process that allows people to cope with brain change, but I think we have to think of many, many other potential mechanisms besides the kind of compensation we can measure in fMRI studies. But that's sort of the beginning of using terms in a different way. Here's a paper by Ada and Prashanthi Brahmuri. Unfortunately, Ada couldn't be here today. She was supposed to be in there. And they introduced these terms resistance and resilience, and they did them specifically to think about pathologies such as amyloid or tau, where resistance would be avoiding the pathology and resilience would be resilience following the pathology. But underlying the terms of resistance and resilience, if you look at this table in their paper, they really did include the idea that resilience is sort of what people are talking about when they talk about cognitive reserve. They just said it's much simpler to call it resilience for the purpose of pathologic studies. And then here's a paper by Tom Montine and crew, where now reserve capacity is really sort of an intercept. There is some injury. They don't talk about brain maintenance. The resistance is not developing lesions, and the resilience is this process of coping with lesions. So resilience and reserve are two different things. And I've seen this model used in animal studies now. I think that people are using these words in different ways. We used in the white paper, we use resilience as an overarching term. Some people are using resilience like one might use cognitive reserve, and take cognitive reserve and make it an intercept, et cetera, et cetera. We just need to come to some closure on these things. So the collaboratory is starting to do this. I think the progress is very, in certain ways good, but in certain ways frustrating. The very first workshop featured 24 different people talking about their operational definitions for these concepts. And you can go to this website, for those of you who aren't familiar with it, reserveandresilience.com. All of the talks are up there, both the videos and the slides. And there was a huge amount of discussion about these concepts. Of course, no closure, but I didn't think we'd get any. So what we decided, we started these work groups, a data sharing work group, a non-human research group work group, a pilot study committee, peer support groups. The pilot study committee, eventually we came to this idea, well, let's give out some pilot awards to both human and non-human researchers, and let's look at the kind of operational definitions that they use to instantiate a cognitive reserve and brain maintenance. And so the second meeting featured 12 pilot award winners presenting their planned research. And again, there was a lot of discussion about these operational definitions. And the third workshop takes place in October 31st, 2021. Hope you can all be there. We plan to have it in Bethesda, God willing, that'll really happen. I think in today's world, we'll probably end up streaming it as well. And then, so let me just give you one little snapshot of this and then I'll stop. So in the white paper, and I made this point earlier, we said this in the guidelines. Research aimed at further elucidating cognitive reserve requires the inclusion of three components: the status of the brain, clinical or cognitive performance outcomes, and a measure of reserve, proxy or a functional measure that moderates between them. So recently I had a meeting with a group of, 10, 12 non-human researchers, and I said, can we start with this? Can we start with this? And the discussion was really very interesting. Well, first of all, I should say that these look like they're cross-sectional. It should really be longitudinal change in status, change in clinical measures, and a measure of reserve. But beyond that, what does status of the brain really mean? Now, for non-human researchers, status of the brain is a very complex thing. So here's sort of a work in progress. It's not official, but this is what I came up with following that meeting with just some of the investigators. So what is the status of the organism, and particularly the brain? Well, it should reflect measures that theoretically are known to be associated with cognition and/or aging. In other words, we can characterize across many dimensions, genetically, in terms of neurodegeneration, et cetera, the brain. So then we can think about a change in some aspects of that status over time, a measure of change in cognition, and a hypothesized moderator. So I'm just giving you a sense of where we want to get, and our hope is before we have this next meeting in Bethesda to involve everybody that was attended this collaboratory and anyone who else is interested in finalizing this part, and then from there we go to operational definitions. So if people agree with this process, I don't really care. If they talk about this hypothesized moderator as resilience, that's fine with me. I would call it cognitive reserve, but at least we all know we're playing the same game. So I hope I gave you sort of an inside look into what's less of a scientific process than the sociology of science, but that's where science really is. These concepts have to emerge and people have to really come to closure on how to think of them. And they're not simple. So reserve and resilience concepts capture important mechanisms associated with successful aging. And of course, that includes disease. Accepted consistent definitions for concepts such as cognitive reserve, brain maintenance, and resilience will be extremely helpful. Similarly, consensus on research paradigms is needed. Influencing reserve and resilience could delay or reverse the effects of aging or brain pathology. And careful, focused research will help us maximize the chance for successful intervention. So that's my hope. I think these concepts are powerful ones. I think that when we get to agreement on how to play this game so that everyone, both human and non-human investigators and epidemiologists and imagers and people who look at hippocampal inputs and circuits on a fine-grained level, we could really come to greater understanding of this concept. So I'll stop there with my talk. It's my pleasure to introduce the next speaker in this very nice series. So this is Lars Nyberg, and he's going to speak about brain maintenance, a key to preserve memory in old age. Lars, we're really looking forward to your talk.
Thank you, Yaakov, for that introduction. When I was listening to you, a memory came to mind from when I did my postdoc with Endel Tulving. So Endel said, a scientist would rather use another scientist's concept, toothbrush, than her or his concept. So I think this is what we're seeing here today. So before we get started, I also like to congratulate the Barcelona team on this great initiative. It's really a fantastic study you're doing, and it was really nice to see yesterday that so good results are coming out. So great work. It will be clearly a contribution to the field. So next slide, please. So as you know, when we get older, some memories start to fade, but not all. If you know how to play an instrument, play football, various cognitive activities, or the facts you learned when you were in school, these do not age very fast. You will retain these memories quite well. But for other forms of memory, next, like these, remembering what to buy when you go to the store, remembering the names of people, when you met, and where you parked the car. These are the age-sensitive forms of memory. So next. These forms of memory then, to remember what you did, say where you parked your car, when you met someone, the what, when, and where, is what Endel Tulving referred to as episodic memory, so memory of your personally experienced events. And this tends to be a very age-sensitive form of memory. Next. So we have been looking at this for quite some time in the Betla study and done this for some 30 years. So we have longitudinal data. And on average, next, we see that episodic memory tend to go down around the age of 60, whereas memory for facts, semantic memory, that is quite stable. It may even improve as we get older and decline much later. So we do see average decline in episodic memory around age 60. Next. Next slide, please. But we also see massive heterogeneity in the aging of people's memory. So next. Here you see this spaghetti graph with trajectories for different individuals who have been part of our longitudinal study for, say, 25 years. And you may see a trend there, but we have used various statistical models to decompose these trends. So next. And by considering the intercept, how well people do when they enter the study, and the slope, how fast they change, we can classify individuals into those who have an average decline in memory, a faster than average, the decliners, and those who maintain memory quite well. So next. And when we pool this over our different age groups, we can see that we have this average group where episodic memory start to decline at around age 60. We have the red group of decliners that start out at the lower level and then show accelerated forgetting. And we have the green ones, the maintainers, who start out at the good level, and 20, 25 years later, they are still at this high level. So clearly then, aging doesn't equal decline in episodic memory. Some maintain memory quite well. Next, please. And here you see the rough proportions of individuals falling into each category. About 2 in 3 fall in the average group, less in the decliner group, but almost about 1 in 5 individuals in our population-based sample fall into the category of maintainers. So these aren't extremely rare people, but you may miss them if you don't sample from the general population, but instead study people who come to a memory clinic. Next. And it's not only us who find this type of heterogeneity in memory aging. It's been shown by the ABC group and also using data from the ADNI cohort, very similar profiles. So it seems to be a robust pattern that you can have a proportion of individuals in the population that show memory maintenance. Next. So the question then that I will focus on here today, and Yaakov already introduced it very nicely, is how we can explain well-preserved episodic memory in aging. So next. This is one model from a paper I wrote with Sara Pudas on successful memory aging. We may have pathological memory decline, we may have average, or we may have successful memory aging, like these maintainers. Next, please. And here is a figure from the paper that Yaakov highlighted, a consensus paper that can be debated, but there too we have reserve, compensation, and maintenance and as key concepts. Yaakov already talked quite a bit about reserve, so next I will focus on maintenance in my presentation, so that we can have individual differences in how much age-related brain changes an older individual show, and that will have marked impact for how cognition is affected in that individual. Next. So there are certain assumptions that go into the brain maintenance concept, and one is that there should be marked individual differences in cognitive and memory change in aging, and I already showed you some examples. Next. And another assumption is that older individuals should differ widely in the amount of brain changes displayed. And next. And that there should be a relation between these two entities. So if you have less brain change, you should have better preserved cognition. So that is the key assumptions. So I'll show you some examples that speak to assumption 2 and 3. Next. So I'll focus on the hippocampus as it is a key region for episodic memory, but in the end I will discuss this a bit more generally. Next. So here you see hippocampus atrophy according to our longitudinal data, and we seem to see that we have significant atrophy after age 50-60, but you can also again see massive heterogeneity. So some have very well preserved hippocampi. So next. Here are two examples highlighted in yellow. One individual up there has a very large hippocampus and show little change, little atrophy over time, and another one has a smaller hippocampus and show much more atrophy over time. Next. And here you actually see the brains of these two individuals. So the high performing is there on the left, the green person with a well-preserved hippocampus and perhaps brain in general. And to the right, indicated by red, you see the individual with a shrinking hippocampus. Next. So when we relate change in hippocampus volume to episodic memory, we find something like this. So next. If we compare amount of atrophy, longitudinal shrinkage in hippocampus volume, we see that it is much greater for decliners, those who have a fast fading memory than the memory maintainers. Next. And we can also see this in the next panel when we continuously relate brain and memory. Next slide, please. So these data link structural preservation of the hippocampus to memory maintenance. I'll also show you an example of functional preservation in the next slide. So here we use functional MRI. So in the scanner, people are presented with faces and names, and they are to memorize these. And we compare the green maintainers with the still really good group, the average. So next, please. And we find a difference. So the memory maintainers, they activate their hippocampus to a higher degree when they do this encoding task than the average individuals. Next. They actually activate it to a comparable degree to people who are half their age, suggesting then that these memory maintainers have also a highly functional hippocampus that may help them to do new memory encoding. So an example of functional maintenance. Next. We have also used other brain markers to look at brain maintenance, including imaging the dopamine systems with PET, positron emission tomography. We have used raclopride and focused on the dopamine D2 system, and we find that individual differences in D2 binding, both in the hippocampus and in the caudate, actually correlate with individual differences in episodic memory. Next. And when we link these individual differences in dopamine binding to the amount of functional connectivity between the ventral striatum and the hippocampus, we find that in part the dopamine memory relationship is mediated by functional connectivity between the ventral caudate and the hippocampus. So if you have an intact dopamine system, you tend to more effectively use then your hippocampus for episodic memory. Next. So next, please. So Yaakov already indicated that we have these pathological markers of memory aging, and one is tau accumulation that is prevalent in the medial temporal lobes, and this is work by Michael Schöll, working with Bill Jagust, showing a relation between tau load in the medial temporal lobes and episodic memory. So the less tau accumulation you have in the hippocampal region, the better is your episodic memory. Next. And many have also linked individual differences in beta amyloid to memory performance, including this study from the Harvard aging study, where you show that less beta amyloid accumulation tends to go with well-maintained episodic memory. So next. So a question then is, what's the mechanism behind brain maintenance? And this is something Ulman Lindenberger and I addressed in a recent chapter in the latest edition of the Cognitive Neurosciences. And here are three examples of factors that could be affected by effective brain maintenance: to maintain neurons, to maintain non-neural sides, to maintain synaptic integrity, and also non-vascular factors. So I'll briefly give you some example from each category. Next, please. So if you start with maintaining neurons, in at least normal aging, we tend to not have that much neuronal cell death. But the question is, do we have preserved neurogenesis? And some argue that we do, and in particular in the medial temporal lobe concept, preserved neurogenesis has been suggested to be a key to effective hippocampal functioning. Next. As you may know, this is a debated topic, and some studies suggest that we don't have much neurogenesis going on after, say, 20s. Next. But there are also studies suggesting that if you are able to maintain memory throughout life fairly well, that may be linked to effective neurogenesis. So this could be one mechanism underlying memory maintenance. Next, please. And I already talked about synaptic effectivity in relation to neurotransmission, say with dopamine or tau load. Next. Another marker of that has to do with the spine density on the dendrites. The arborization itself may not be that age sensitive, so sort of how rich the dendritic trees are, that's not that much affected in normal aging, but how much spines you have there is clearly influenced by aging and linked to cognitive performance, as shown in this nice review. Finally, vascular factors, they are discussed more and more. And next, in this short review paper that I wrote with Anders Wåhlin recently, we have tried to outline the important vascular events that may lead eventually to declining episodic memory. So you have stiffening in the aorta, in the big arteries in the brain, and in that way, when blood is pumped from the heart to the brain, you may actually have damage to the brain, in particular the pericyte cells that make up the blood-brain barrier. And that could in turn affect how effective the neurovascular coupling is. And if this is not functioning properly, we know it's very hard to effectively distribute nutrition to various parts of the brain, and that may in particular play out into negative effects on demanding episodic memory tests and say working memory tests where you need to sustain neural activity over extended periods of time. So next, please. So I've focused in the presentation on hippocampus maintenance, and this is because we know it's a key structure for episodic memory. But we have also started now to look into how general is this? Does it sort of extend to other brain regions and also to other forms of cognition? So we have looked at that using longitudinal data over a decade, where we have large individual differences in change in our sample. Next, please. So here you see frontotemporal regions, you see superior frontal regions, right parietal regions, and limbic regions. Limbic regions including hippocampus and the parahippocampus. So all of these regions change. They get, we have atrophy in them over 10 years, and we have individual differences in rate of change. We can link them to a general brain factor and also examine it at the specific brain regional level. Next. And we can link that then to episodic memory, but also to speed of processing and how well you perform on a visospatial task, block design. And these in turn can also be integrated into a G measure of general fluid cognition. Next. And we can then analyze the relation between these factors at the overall level, a general brain factor with a general cognitive factor, and also at the level of specific brain regions with specific cognitive functions. And the general message so far in our data is that we find support for specific maintenance, in particular that preserved hippocampus goes with preserved episodic memory. For example, preserved superior frontal integrity is linked to preserved block design performance. So next. So I think in the future, a key question is whether we can promote brain maintenance. As Yaakov showed us in his presentation, there is absolutely no conflict between brain maintenance and reserve. If your maintenance falls, you like to have a good reserve to help you cope with cognitive and everyday life challenges. But still, it will be important to see what we can do as we have an aging population globally. So next, please. So I think we can consider a number of different factors that could potentially promote brain maintenance. And next. One that we have looked at that seem to characterize the memory maintainers is physical activity as of now and how physically active you have been over time. Next. And in recent work on the brain age gap, we also found in work by Tora Dunås that those people who had sort of a youth-like brain, they were also more physically active. So both for memory maintenance and brain maintenance, this seems to be one example of an important factor. Next. So I think we can look at many more potentially, and many that will be discussed and have been discussed at this particular meeting. So next. I think we have learned a lot. And again, congratulations to the Barcelona Brain Health Initiative. It's great, and I think it will be important to learn more about nutrition, sleep, and other factors that have been discussed. So on that, thank you very much. And it was a challenge. I didn't see my slides, but I hope you saw them better than I did. So thank you.
That was fantastic, Lars, especially if you didn't see your slides. That was a great talk. Thank you so much. I should remind the listeners that there will be discussion at the end. There's a place for you, I assume, to put in your questions. We'll be getting a stream of those, and we'll be asking them at the end. Okay, so now it's my pleasure to introduce Gaël Chételat from France. Her talk is Multimodal Neuroimaging and Lifestyle and Aging and Dementia. And I'll warn you, Gaël, that before you begin, there's a little brief video.
Okay. Good afternoon, everyone. Thanks a lot for the invitation. I would like also to congratulate the Barcelona team and initiative and thank them for their invitation. So I will be talking about lifestyle-based intervention in brain aging and dementia, and more particularly about meditation training and the impact of meditation training on mental health and well-being in aging population and the preventing dementia. I will present a hypothesis that we have developed with the Meditating Consortium funded by the European Committee, and that has been more particularly fueled by the expertise of Antoine Lutz, expert in neuroscience of meditation working in Lyon. Our model proposed the hypothesis that mindfulness meditation combined with loving kindness and compassion meditation in the aging population could constitute a protective lifestyle, not only on Alzheimer's disease, but also more generally on cognition, mental health, and well-being. We argue that these meditation practices would operate through both attentional control, metacognitive monitoring, and prosocial capacities promotion that would positively affect aging. And these mechanisms are thought to have a positive impact through two mechanisms. First, by promoting positive, favorable, beneficial, positive factors on aging. And second, by down-regulating adverse and detrimental negative factors. So I will develop a little bit about that later on. First, a few definitions. We define meditation as a family of complex emotional and attentional regulatory strategies, which goal is various, but include the cultivation of well-being and emotional balance. Secular mindfulness-based meditation is most often used in scientific research, and I would really discuss about secular practices. More specifically, among this family of practices, we have mindfulness meditation that we define as the practice to cultivate a vigilant awareness of one's own thoughts, actions, emotions, sensations, motivations, and directly target attention and emotion regulation ability, which is particularly relevant in the context of aging, we think. Loving kindness and compassion meditation refers to the capacity to cultivate benevolence, acceptation, and care for oneself and for others. So we have already discussed a number of risk factors that can be modified. So lifestyle factors that can be modified can allow to prevent a lot of cases with dementia. For example, this study from Barcelona has shown that with 10 in gray or 25% reduction of the main risk factors, including low education, smoking, physical inactivity, depression, hypertension, diabetes, etc., we can reduce by a high million of cases and prevent the number of cases worldwide. So this is a very important thing where we can really act on Alzheimer's disease. This is a more recent publication showing that especially in late life, the factors that are the most relevant are smoking, depression, physical inactivity, social isolation, and diabetes. And these are the factors that are particularly relevant as we are getting older. Based on this knowledge, there have been large-scale multi-domain, long-term, non-pharmacological intervention that has been launched. I give a few examples of the largest worldwide initiatives here. And just taking this example, the finger study here, in this study, you can see here they combine diets, intervention, physical activity, cognitive training, as well as group management of metabolic and vascular risk factors with a visit from a nurse that give instructions. So they combine all those in a trial intervention. And you can see that this trial target the main risk factors that I've mentioned before, except those relating to depression and marginally to psychoaffective factors such as stress, anxiety, sleep difficulties, social isolation. These factors have been shown to have a important impact on the risk for Alzheimer's disease. As you can see here, the risk increase as depression increase in the aging population. And you can see here that cognition decreased over time more steeply in participants with depression compared to those with lower level of depression or no depression. This is a more recent study that you probably already heard about yesterday by Natalie Marsh, showing that repetitive negative thinking, which is also called as perseverative cognition, and is defined as a behaviorally measurable process that encompasses future, but also past directed thoughts, including both worry and rumination. And these factors was related to the two main neuropathological landmarks of Alzheimer's disease, including both amyloid deposition and tau pathology as measured with PET in the brain. So the higher score you have of repetitive negative thinking, the higher level of amyloid and tau deposition you have. It's the same for anxiety. You can see here the increased cumulative incidence of dementia in people with higher anxiety compared to those with a low level of anxiety. And stress is also considered as risk factors for aging, notably through mechanisms including stress hormones having a negative impact on the hippocampus and cell proliferation and neurogenesis, etc. So that chronic and high level of chronic stress might have a negative impact and be risk factors for, notably for Alzheimer's disease. Okay, now let's talk about meditation and why we think that meditation might have a positive impact and which are the assumed mechanisms of action of meditation on aging. And these are the basis for our specific hypothesis. Several models have been proposed as for the key mechanisms relating mindfulness meditation to different types of training and to neural reserve, and notably, as you can see here, both the attentional system and the multi-domain system are thought to be involved. Here, the novelty of our model is to first integrate both mindfulness meditation and loving kindness and compassion meditation to promote healthy aging. I will just develop a little bit to explain why we think those two approaches are complementary and promote each other's towards promoting healthy aging. Second, we also, in our model, we also integrate the fact that it is relevant, important to study meditation interventions at multiple time scales, spanning across several years to identify the different time courses and changes induced by meditation across the psychological, neural, and biological markers of aging, as we think that according to the outcome, the endpoint we are interested in, we focus on the time scale is differentially relevant. So the rationale for previous hypotheses that have proposed that mindfulness meditation are relevant for mindfulness meditation is relevant for aging, and the rationale for adding loving kindness and compassion meditation is that studies have shown the positive outcome of specifically loving kindness and compassion meditation on mood disorders. Also, LKCM has been shown to down-regulate social stress with different mechanisms than mindfulness meditation. And you can see here that the different practices differentially impact the different scales that are measured here and have different impact on subjective experience, the HPA axis, the autonomic nervous system, and the innate immune system, suggesting that as they operate through different mechanisms, mindfulness meditation, as illustrated here in yellow with the presence, versus loving kindness meditation, represented here as affect, would operate through complementary mechanisms. Third, LKCM is for loving kindness and compassion meditation, is thought to increase positive emotions and purpose in life. And it is also thought to be mediated by different cognitive and neural mechanisms than mindfulness meditation. There are only very few studies that have specifically investigated in a very reliable design LKCM versus mindfulness meditation. And the most knowledge we have come from to date come from the big Rousseau's project conducted by Tanya Singer its collaborators and described in this publication. And you have here one of the publications from this project, and they directly compare different kinds of meditation practice. You have here the core exercises that are involved in each of those three practices. You have the presence module, the affect module, and the perspective module. And you can see that those different modules involve differentially the attentional component, the compassion component, and the theory of mind components of that are thought to be involved in meditation. So the mindfulness meditation that we refer to more specifically refer to the presence module in yellow, while the LKCM meditation practice more specifically refer to the affect module. And you can see here that the presence module mainly involve attention, while the affect module mainly involve compassion. You can see here that in this study they showed that there was specific correlation and module-specific changes in cortical thickness and specifically increase in cortical thickness related to the different modules. And these show that the brain regions that are more specifically involved in one practice is different from brain regions involved in the other practice. You can see here in the presence mindfulness practice, you mainly involve the medial frontal and also other prefrontal regions here, as well as more posterior areas, while in the affect module, you mainly have the cingulate cortex, the insula, and the temporal parietal regions. And those are differentially correlated with performances as illustrated here. So in this model, we propose that LKCM and mindfulness meditation operate through upregulation of positive and protective factors for aging and downregulation of habits that have a negative impact on the brain. Those positive impacts would operate through the upregulation of automatic positive schema, such as caring and altruistic behaviors, while mindfulness meditation would more particularly down-regulate automatic negative schema. For example, rumination that I have mentioned before, repetitive negative thinking, conflict avoidance, and resistance. Our model also proposed that those two mechanisms would involve different brain regions based on the results I presented to you before, but also on meta-analysis of for example, Fox and collaborator have performed meta-analysis where he showed the main brain regions involved in meditation and different meditation practices. And we think that mindfulness meditation might more particularly involve the medial ventral prefrontal cortex and hippocampus. So this is for mindfulness meditation. Loving kindness and compassion meditation would more particularly involve the insula, temporal-parietal region, and entorhinal cortex. And some other regions are indifferentially impacted by both mindfulness and loving kindness and compassion meditation. As here, for example, the posterior cingulate, the more really ventral part of prefrontal cortex, and internal part of the temporal-parietal regions. So as a whole, we think that those two practices combined would have would upregulate favorable beneficial positive factors, and this include cognitive and brain reserve if we separate them, cognitive stimulation and activities, social interactions, well-being and happiness, and some studies have shown a positive impact on the telomerase activity and telomere length. And we think that they would also operate through down-regulation of adverse detrimental negative factors, and these include cardiovascular risk factors, depression, sleep disturbances, stress, anxiety, cognitive decline, and frailty and disability. We think that altogether they would have a positive impact on aging. And we think we would be able to test this hypothesis with the European project called Silver Santé Study, or Meditaging, Silver Santé Study being the public name, where our goal is to investigate the impact of meditation training on mental health and well-being in aging population. This is our consortium. Antoine Luce, I mentioned before, which is our expert for the meditation work package, Julie Gonneau, postdoc in my team, who is responsible for the lifestyle work package, Fabienne Colette and Olga Klimecki in Belgium and Switzerland for the attention and emotion work packages, so to test the mechanisms of the interventions, and Nathalie Marchand, who you have heard about earlier in this meeting, responsible for cognition and well-being in my team for the biological markers. So I don't have much time to detail here. I just want to mention that we have two very large clinical trials. One is on SCD population, subjective cognitive decline, where we are interested in assessing the impact of a short meditation intervention, 8 weeks, based on MBSR practices, so mindfulness-based stress reduction practices, in patients with subjective cognitive decline with both behavioral and blood outcomes. And this study has already shown some results on anxiety, which is our main outcome measure, which is ongoing for the publication, so not published yet. And we showed a positive impact of both the meditation intervention and the health education program on the measure of anxiety, and this effect was maintained after 6 months. And the second clinical trial was conducted on cognitively intact elders. It involved here both behavioral and biological measures, being blood sample, but also multiple neuroimaging measures, and a very detailed assessment of sleep. People had been randomized, and they had an 18-month intervention with both an active control, which is learning English, English lessons, and no intervention, so a passive control. The intervention include both mindfulness and compassion, as has been discussed before. So we think we will be able to test our hypothesis. So this is all our contributors, and thank you very much for your attention.
Thanks so much, Gaël. That was a great talk. I want to remind everyone that they can, to the right of the panel where you're seeing the talks, there's a Q&A box. Please feel free to enter questions about any of the talks as we go on, and then we'll recapitulate your questions at the end of the at the end of the final talk. So regarding the final talk, it's my pleasure to introduce Alex Iranzo from Barcelona, who's going to speak to us about sleep. Alex, you just wait for the little video before your talk starts.
Okay, thank you. So I would like to say thank you very much to Álvaro Pascual-Leone, David Bartres, and Antonio Esteve for inviting me to give this talk about the sleep. As you can see, the title is very short. It's the shortest title that I have seen in my life from my own perspective, but the topic is huge. But I'll try to do my best. So the outline of this presentation is that we're going to talk first about the sleep and its function, second about the effects of not sleeping at night, or sleeping bad, or sleeping small quantity, and also a little a brief talk about the sleep and neurological diseases, their link. So sleep and its function. As you probably know, sleep, we can define sleep as a recurring and cycle state, which is universal in the animal kingdom, is essential for life, and is regulated by the brain. In fact, during wakefulness, there are a number of structures and neurotransmitters that are connected between themselves that produce that we are completely awake, while when we are asleep, a different number of structures or neurotransmitters are activated and others are inhibited. If we perform a PET scan evaluating the level of glucose in the brain, we can see that in wakefulness there is a high activation of the whole brain. And when we are asleep in what we call slow wave sleep, I mean, there's much less activation in the brain. And then when we are asleep in REM sleep, the activation is higher, not as equal as during wakefulness, but it's also higher and other structures are activated. The transition between to be awake and asleep is not rapid. I mean, it occurs in few seconds or minutes. And this is there's a switch, or what we call a flip-flop system, where some neurotransmitters and structures in the brain are activators and others are inhibited while we are awake. And the opposite, the other side of the coin, comes when we are asleep. That GABA, for example, is activated, and others like noradrenaline or serotonin are inhibited. Well, the sleep architecture of a young adult, we can divide the structure of the subject in two different stages. What we call non-REM sleep, which is 75% of the night, and REM sleep, which is 25% of the night. And we can divide the non-REM sleep in what we call light sleep. Stage NI1 is what we call drowsiness. It's a transition between wakefulness and sleep. Then we are right to stage 2, and then we are right to stage 3. Different percentages of these stages across the night. Well, sleep is a continuous process over the night, but also it occurs in different stages. So at the beginning, when we are falling asleep, we are in bed, for example, we're trying to fall asleep, and then we reach drowsiness, stage 2, stage 3, then goes to stage 2, and then to REM sleep. And the first REM sleep occurs around the 60 and 90 minutes after you have fallen asleep. And there are like 5 different cycles, between 3 and 5 different cycles across the night. So in the beginning, in the order, the first third of the night predominates slow wave sleep, what we call N3, and in the last third of the night predominates REM sleep. This is why, for example, the parasomnias that occur during slow wave sleep, for example, somnambulism, sleepwalking, they occur in the first third of the night, while other parasomnias, like for example, REM sleep behavior disorder, the abnormal behaviors occur during the 4 a.m., 5 a.m., 6 a.m., and so on. Well, how sleep is modulated? Mainly or basically is modulated by 4 different systems. homeostatic system. That means that when you are more awake, is the more you need to be asleep, or the opposite. The more you are asleep, the more you need to be awake. Also, there's a circadian system, ruled by the circadian clock, which is in the hypothalamus, and we all fall asleep. We humans fall asleep according when it's low light. For example, during darkness, we need a specific temperature and the time of the day. There's a rule of number 3, that during the day, the maximal sleepiness of a normal person is at 3 p.m. and at night is at 3 a.m. Also, there are some individual differences that regulate sleep. For example, a newborn may sleep like 16 hours per day, while people in the 50s usually sleep between 7 and 8 hours. There's a mild difference between gender. Usually women sleep with more slow wave sleep than men. And also there are external factors. It's impossible to sleep with a discotheque under your feet, or when you have anxiety, or you are predisposed to fall asleep in a boring meeting at 3 p.m. when you are bored. Like I said, there are different between the species. For example, we humans are awake during the day and asleep during the night, and the opposite comes to the rodents, that they are asleep during the day and they are awake during the night. As you can see here, all species, like I said, are awake and asleep during a whole day. And we have some animals that sleep more than 3 hours in REM sleep and others in non-REM sleep. For example, an adult sleeps around 8 hours average per day, and 2 of them are spent in REM sleep, while the dolphins sleep 10 hours and they sleep like 25 minutes during REM sleep. And the dolphins are really peculiar, because when they are asleep, half of the brain, one hemisphere, is asleep and the other is completely awake. And the opposite comes after 30 minutes, because they alternate the hemispheres to be awake and asleep. This is quite peculiar. And also, if you travel to Japan, this is a typical picture, not to see only adolescents, but also all kind of people are falling asleep during the underground. And this is typical because I think that people from Asia have a predisposition to fall asleep in normal circumstances. Well, what is the role of sleep? Why do we sleep? It will be a mistake if there is no role of sleep, because we're sleeping around 7, 8, 9 hours per day. So it will be a mistake if sleep has no role. So basically, why do we sleep? Because we are restoring our brain. There's a metabolic restoration and maintenance during the whole sleep, and during this process there's a huge cortical plasticity, synapses stability, receptor recovery, and protein clearance, such as, for example, amyloid. And you know that it's been published that there is a correlation between sleeping less and accumulation of amyloid in the brain that may link sleep with Alzheimer's disease. Also, during sleep, we have a memory and learning consolidation. There's a huge information that is clear in the hippocampus, emotional and mood control, immunologic stability, and endocrine regulation. This is the function of sleep. And also creativity. For example, Paul McCartney dreamt the melody of Yesterday, and then the next morning he started to write the words, and he wrote Yesterday with the working title of Scrambled Eggs, because he was having breakfast scrambled eggs. So what are the effects of sleep deprivation, which is the other side of the coin? Well, what is the origin of sleep deprivation? We have two different causes. First is sleep a very small amount of time during the night. And this could be because you have insomnia and because you decide to work at night to expand your time awake working, or because at night you are with a tablet or with a mobile, and this incredible light in front of your eyes go to your brain and and it's impossible to sleep like this or later on. And also because of poor nighttime quality. The quantity is okay, but the sleep is fragmented. For example, if you have a high amount of sleep apneas that fragment sleep. So what is the amount of nighttime sleep? So this is a graph, a cartoon that shows the percentage of people sleeping less than 7 hours, and that increases from 1985 in light blue to red to 2004. And you can see that in all ages, the amount of sleeping less than 7 hours increase across the lifespan. So we are sleeping less. What are the effects of sleep deprivation? Both acute and chronic. So it's lower cognitive and performance, poor memory, less attention and concentration, increased daytime sleepiness, irritability, anxiety, low mood, fatigue, less creativity and productivity, and decreased libido. So nothing good of all these characteristics. They are all not very good, they are bad. And then what are the effects of sleep deprivation? So you are making errors, you're taking wrong decisions or making bad plans. You are prone to have personal conflicts, having economical consequence, abuse of stimulants, hypnotics, alcohol, or drugs, an increased proportion of traffic accidents, and predisposition of some diseases. What kind of diseases have been linked to a poor sleep or chronic sleep deprivation? So important diseases, in fact: coronary heart disease, stroke, obesity, hypertension, diabetes type 2, depression, Alzheimer disease, some types of cancer, and upper airways infections. So overall, decreased sleep quantity and quality produce less economical productivity, creativity, and less health. So the message here is that you need to sleep well and enough to be more happy, to be more productive at your work, with your friends, with your family, and to be more healthy. Let's try to link neurology and sleep. So, basically, there are huge books like this on sleep and neurology. I'll try to summarize in 2 or 3 minutes. So all kinds of diseases have been linked to abnormal sleep or sleep abnormalities or sleep disorders. Alzheimer's disease, Parkinson's disease, stroke, multiple sclerosis, anti-IGLON5 disease, epilepsy, amyotrophic lateral sclerosis, and Resilence syndrome. And in one way or another, they have been linked to insomnia, hypersomnia, abnormal sleep patterns, and parasomnia. So neurologists, as medical doctors, have to be aware that sleep disorders can be a crucial complaint or important complaint of our patients with all kinds of neurological diseases. Let me focus on one particular disorder, one particular sleep disorder, which is called REM sleep behavior disorder, that our group in Barcelona during the last 15 years have focused our attention on this disorder. Oh, this is parasomnia. This looks like a somnambulism or sleepwalking, but not in the adolescent or in children. This occurs in people older than 50 years. Mainly, what I can tell you is that during REM sleep, you are asleep and you are dreaming, but you are paralyzed because your lower brainstem is inhibiting your spinal cord. So if you dream that you are typing, you are not doing like this. If you are dreaming that you are fighting, you are not punching. If you are dreaming that you are singing, you are quiet. This is what occurs in normal people. But in REM sleep behavior disorder, the patient have nightmares, typically the same. They have been chased in the nightmares, or they have been fighting in self-defense, and they have a dysfunction in the brainstem. So the brainstem doesn't work well, And when the patient is dreaming about these unpleasant dreams, they are displaying vigorous dreaming active behaviors because they shout, they yell, they punch, they knock off the nightstand, they fall asleep. And this looks like a parasomnia, but this occurs in REM sleep. And then the diagnosis, you have to perform a polysomnogram, and then you see that in REM sleep, there's an increased electromyographic activity. And in the synchronized video, you can see how the patient is punching or shouting. This is REM sleep behavior disorder. Okay, nice. This looks like a parasomnia and a sleep disorder and so on. But we have, but the second part of the story is that we have followed these patients and we have seen that REM sleep behavior disorder, this sleep disorder, represents the first manifestation of Parkinson's disease before the onset of tremor in the hands and a slow body movement. So these patients that REM sleep behavior disorder come to us without neurological complaints, without cognitive complaints. The brain MRI is completely normal, and the neurological examination is completely normal. But if you follow these patients close from the clinical point of view, you will see that sooner or later, you, as a doctor, as a neurologist, you will diagnose all these guys sooner or later with Parkinson's disease. After the diagnosis of REM sleep behavior disorder, after 5 years of follow-up, 1/3 have been converted to Parkinson's disease. They have developed tremor and bradykinesia. After 10 years is 76%. After 14 years is 91%. So almost everyone will be diagnosed with Parkinson's disease. So you have an opportunity here to do something and try to prevent Parkinson's disease. As you know, Parkinson's disease is characterized in the brain because in some structures of the brain you will see neurodegeneration. That means that there is a excuse me, neuronal loss. The cells are lost and damaged. They are being killed. And those that they remain alive, they have what we call Lewy bodies, that they are some inclusions with alpha-synuclein, which is a protein. So we have just performed lumbar punctures in our patients with idiopathic RBD, idiopathic REM behavior disorder, and we have found that in 90% of these falls, without any complaint as neurological complaint as motor or cognitive, they have alpha-synuclein, the devil, the enemy. So the hallmark, the pathological hallmark of Parkinson's disease. And this has been accepted 3 weeks ago by Lars, a neurologist. So it's the first time that in a meeting I'm free to say this finding. So what we are facing right now is that from one sleep disorder, you can predict the evolution to Parkinson's disease. And this is very important because in people with REM sleep behavior disorder, we can try to make a neuroprotective drug assessment against, for example, alpha-synuclein, because now we have some immunotherapy that may prevent the onset of Parkinson's disease. This is what we call neuroprotection. And this is just an example how from one sleep disorder you can link to neurology. Thank you very much for your attention.
Thank you so much for that very nice talk. I'd say, unfortunately, I missed the beginning yesterday morning, but I think because it was 2 o'clock in the morning for me, I'm not that dedicated, but I see that throughout the entire meeting, we focused on a lot of different potential influences on successful aging, psychological factors, environmental, various games or digital interventions, nutrition, exercise, social relations, cognitive training, today meditation and sleep. And then we've talked about these concepts of cognitive reserve and brain maintenance. So now we have a bit of time for some questions. I think maybe what we can do, Álvaro, is you and I can take turns reading. You have access to them as well. So the first question that I see is for Lars. What do we know about the relation of structural brain changes or even fMRI changes and EEG metrics of brain activity and maintenance? So I guess the question focuses on EEG.
I'd say not much. If you imply simultaneous recordings, etc., I think perhaps a bit more if one looks at it in isolation. But it's an interesting question in relation to the generality versus specificity of the notion of maintenance. Sure, there could be some EEG waves that are really sort of tagging into general maintenance, and others that we know are more linked to hippocampal processing. So my guess would be some ERP studies on those could potentially give you overlapping findings, what we see for structural hippocampal atrophy and fMRI activation. But I can't think of a study right now that have actually looked at it. So room for an experiment.
Okay. Gaël, a question for you. Is there any evidence of differential effects between different types of meditation, or even ways to define best meditation practice for a given individual?
Yep. So a very good question. So there has been, for example, a meta-analysis published by Fox and collaborators that show the different impact and activation of brain regions and changes in these brain regions according to the types of meditation. And they show that there are some brain regions that are similarly involved, other brain regions that are specifically involved in the one or the other of those different practices, not only mindfulness and loving kindness and compassion, but also other kinds of meditation. However, I should say that the evidence for now are quite young, new, and they are not so strong for now. So more clinical trial with longer term and larger samples are needed to really test for the specific effect of meditation, of specific meditation practices on the brain. One of the objectives of the Meditating Consortium project is really to assess this question, so really understand the different impact on brain, but also cognitive well-being, extra measures, including as well sleep, as we have just discussed. We can see the relevance of sleep in this context, and to also understand the mechanisms so that the possible future intervention can really be more specifically focused, so that to be more efficient, and that the active compound can be promoted.
Okay, thank you. Here's a question.
I can jump in and
go ahead, Álvaro.
Ask one question that was asked for you, Yaakov. There is a question by an anonymous listener who asked whether you could comment on whether something can be done to increase or maintain a cognitive reserve across the lifespan, or is it all defined by education? Can something be done later on?
I think that the epidemiologic evidence is quite clear that there are different features across the lifetime that seem to contribute to more successful aging. And I think some of that is via cognitive reserve. So of course you have education, which occurs usually early in life, but we see people now going back and doing late life education. So social networks are something that might be something important later in life. Leisure activities are throughout the lifespan. So I think it's a lifelong process. The nicest papers I've seen are from, well, there's one paper from Marcus Richards from the British birth cohort, where they actually have information on these people since they were born, and he can show that there are separate paths to cognition in old age, from socioeconomic status, from education, from occupational exposures in midlife, from leisure activities later in life, and each of them contributes separately. So I think it's a lifelong process. And I think that's, of course, the hopeful message. And a lot of these interventions that we're hearing about, or different modalities, have the promise of contributing in different ways throughout the lifespan. Maybe I'll read one. So there was another question to you, Lars. Specific maintenance associations that you mentioned were referred to brain structure cognitions associations. Is that correct? Do you think this would, do you think that would stand also at a functional level?
Well, hard to say, but we have placed our bets on specific maintenance, so I guess I have to say yes, and wait for the data.
Yeah, I would agree. I mean, there was a question addressed to me, if you don't mind, Álvaro. So the person asked me sort of the flip side of the question to you, a different questioner. If cognitive reserve progressively accumulates over a lifetime, would it be correct to hypothesize that cognitive reserve reflects into specific brain structural rather than functional features? So it's an interesting question. Up until now, especially in human imaging research, what we've tried to do is hold structure constant as much as we can and look at different activation, which we believe reflects functional uses. So in other words, you have the same, I would call it hardware, I know that's a bad word, and the software is programmed better to be more flexible. But I think that when we get to non-human research, where we can really get to the absolute networks that underlie cognition, that might not be the case, although there are indications. For example, Carol Barnes can show that animals lose specific inputs, let's say, to the hippocampus, but those inputs become more, in some animals, become more robust. So that would not be a structural implementation, but a functional one. So yeah, I mean, these are deep questions. Álvaro, maybe you pick.
I have a question. I want to convey a question that was posed for Alex Iranzo. The link between sleep and Parkinson's disease is very exciting. What is known about the link to dementia, and can something be done through sleep to prevent decline, age-related decline?
Well, there's a huge information now, especially during the last 5-7 years, regarding dementia or Alzheimer's disease, in fact, and sleep. And mainly, there are 2 sleep disorders that have been linked to dementia, to Alzheimer's disease, that they are in fact insomnia, or sleeping, or shorter sleep during the night, and also obstructive sleep apnea, which is also linked with the deposition of beta amyloid on poor cognitive performance. So that means that trying to improve insomnia and to solve sleep apnea with any kind of therapy, like for example CPAP, may try not to decrease or eliminate Alzheimer's disease from the world, but maybe to improve the quality of these patients, or at least decrease the risk of having dementia. But if you have some genetic background or some familial background, I mean, this is going to be difficult, whatever your sleep is.
Okay.
Thank you.
We're getting there. One last question for you, Gaël. I guess this is based on your findings. Do you think neuroimaging-based information could be used to guide personalized prescriptions of specific meditation-based intervention? So that's something I know that you have ideas about how neuroimaging might be influenced or reflect the impact of meditation and different types of meditation. You have to unmute.
Yeah. Yes, this is a good question. We also have an experiment in expert meditators, so that people who have 65 years old or more who have practiced all over their life different types of meditation are practicing meditation during the scan, so that the objective here is to identify signatures, brain signatures of different types of meditation. And we think we could use that to further understand which of the specific meditation compound have particularly had an impact on our aging population that were naive for meditation and have then progressed during the 18-month intervention. So using all those information together, we would first understand the mechanism of medication in the whole sample. We think that we might be able to maybe, to answer more directly to the question, maybe target more specific subtype of the population that might more particularly be sensitive to one type of meditation or the other. I would just add that those two kinds of meditation are thought to be not only complementary, but also to constrict from one to the other. So that, for example, first practicing mindfulness meditation might help to then be able to practice more specifically loving kindness and other types of meditation. So that would be kind of lowering or hiring some kinds of meditation practices, but we probably, our hypothesis is that both combined is better, then we can adjust the level of each.
Okay, thank you. So I think we're reaching the end of the time. I would like to thank the organizers for inviting me to be part of such an interesting set of talks. And I'll leave it to Álvaro to wrap up.
Thank you very much. Thank you all for this fantastic session. I think what we hopefully have been able to do, thanks to all of you and from yesterday and today, is provide a bit of an overview, a forum to address some of the factors that there is evidence can contribute to brain health along the lifespan, and specifically to highlight the fact today that some of these factors can work in different ways. It can work by promoting healthy aging and maintaining a function. They can work by modifying the influence of pathology. They can work by impacting reserve. And that conceptual framing of these issues is important, but that we're also at a point where we can maybe continue to gain insights while starting to translate some of those insights into specific recommendations. And the risk of putting you all at the spot, let me finish with some question that was posed yesterday to the panelists in one of the other sessions on the lifestyles, and that I think be a nice one to maybe close on, which is, are we actually at the point where from a clinical perspective, physicians like myself could start or should start not just generally recommending, but actually prescribing and supporting the adherence of lifestyles, the recommendation of meditation, or the focus on sleep as a means to actually maintain brain health, maintain cognitive ability, and continue to build on reserve. Are we there where we need to face a sort of more holistic lifestyle emphasis rather than just simply diagnostic and medication-based therapeutics? Now, I'll just leave it for all of you to comment if you would like to share some thoughts.
So maybe I'll start. I think we are there. I think that there's enough evidence, when you see these reviews, Gaël quoted one, there's multiple reviews of lifestyle, various features that are associated with dementia, and these lifestyle factors are always in those reviews based on epidemiology. I think the hard part is how careful we can be in our statements, how definite we can be in our statements. And another thing that I've always been concerned about, I'll raise it, is this happened in cancer research quite a while ago, where this positive state of mind was related to better outcomes in cancer. And then you have almost the person blaming themselves. Well, I have this cancer, I probably didn't do it right. So I think we have to be very careful about that, especially for aging, it's one thing, but for Alzheimer's disease, it's a horrible disease that really, I think once it begins, is hard to stop, and it's going to win. But I think beyond that, when people ask me, I say, look, the evidence is very strong for physical activity, for healthy diet, for maintaining activity and interests, not anything in particular, but doing what you want to do, for meditation, or Tai Chi, or other meditative practices. I think that's as far as we can go. I think to prescribe a specific game or a specific activity, but I think that encouraging that activity is correct medically and scientifically.
I will continue maybe. Thanks, Yaakov. I fully agree. I think that if we are here, I'm also, I agree with Yaakov, we probably need to, it depends from which intervention we are speaking about. Maybe we have more data for physical activity, for example, that for now, meditation practice, we need more insightful, more input about the dose, in addition to the type of practice. I think that people in general are very happy to take activity, to take actively their health in charge. So I think this is a good point that should be taken into account. And finally, I think what we need to better understand is which intervention is best according to people. There is their motivation to be taken into account, but also their profile being genetic, as had been briefly mentioned, but also maybe sex and maybe other factors that might change or modulate the impact of the different interventions.
I can only add that I think the question is interesting, whether a physician should use it, and I agree with Gaël and Yaakov what they said. But from a brain maintenance perspective, I would also like to push the question a bit by saying that it's not about the time when you go to a physician, it's about primary prevention. It's what we do when we are young, in middle-aged, and so on. So it's kind of a sort of a societal awareness that you can, to some degree, I mean, we know that the genetic factors are huge, but to some degree, there is evidence that you can impact your aging, your risk for disease, etc. So I think it starts there. Then, of course, a super big question is also, we have knowledge that is solid in many areas. Why don't we stick to them? So sort of we can have perhaps short-term behavior change, but the long-term is super interesting and hard and may tap into personality and these type of issues. So yeah, we can prescribe, but we should start earlier.
Thank you so much. Alex, I don't know if you have any comments.
No, there's little to add after these wonderful interventions from the speakers here in the panel. Well, regarding sleep, I mean, it's pretty obvious that we always thought that to sleep it was a waste of time, and I prefer to be more awake to. Oh, how good! I only slept 4 hours today, or 5 That means I have 24 hours to be productive. Well, it's not the case. It's not the case. We have learned that is not the case, and that we have to promote the sleep as we are promoting during the last 5-10 years, exercise and diet. And that means that we have to change our habits and our lifestyle, and to think about that sleep is important, and to go to a sleep doctor or to the GP if you have any problem like insomnia or sleepiness, or you have a bad habit. So this is very important from the beginning of your life, especially now for adolescents, that they take the mobile at night, and they still have the mobile on in the middle of the night. And if they awake in the middle of the night to go to the toilet, they switch on the cell phone, and if there are some messages, then they start to write and respond, and this is a mess. So from the beginning, from the adolescents, we have to learn we have to teach our kids or our population that the sleep is important, and having regular habits, it's crucial. It will be crucial.
Thank you so very much. That's a perfect sort of maybe point to end. And I want to thank you all again for the session today. And more broadly, we hope that that the message of the need for a bit of a sort of transformative approach, where we emphasize a lifelong emphasis on brain health rather than try to catch up with devastating illnesses when and if they present, is important. There is reason to hope in the case of the illnesses, but there is a need for a broader effort of a sort of ecosystem that goes through education of individuals, to targeting personalized interventions of prevention, but also to public health efforts of primary prevention and policy. And that sort of an ecosystem requires awareness that has to be grounded in science. And we really appreciate the support from BioCAD, EIT Health, Guttmann Institute, La Caixa, Universidad of Barcelona, and all the speakers for allowing us to hopefully start putting some light on that and contributing to the effort. I think it was particularly timing that the United Nations yesterday established the decade of healthy aging as the focus of the next decade. We are enthusiastic supporters of that, and we hope that we can build on this workshop to carry it forward and contribute to that important goal. So thank you very much on behalf of David Bartres and Chema Tormos and the Barcelona Brain Health Initiative. We are honored to have been here, and hope that you found it interesting.
It's been a pleasure. Thank you very much.
Thank you.
Thank you very much. Bye.
Thank you.